Session 2.3.4 Analytic Experimental Designs Deck 9 Learning Outcomes 1. Describe the experimental study design. 2. Identify the key elements in an experimental study. 3. Understand the process of designing a randomized controlled trial. 4. Define and differentiate the types of trials. 5. Discuss the strengths and weaknesses of experimental studies. Introduction. What is epidemiology? Relationship between X and Y. Ask AI. Alt. Experimental versus observational studies. Experimentation. Exposure is directed, or assigned, by the researcher. Observation. Exposure is observed by the researcher as it plays out over time. Confounding. Confounding is most simply defined as the association between an exposure, an outcome, and a third extraneous variable known as a confounder. Z as the confounder in this diagram. Ask AI. Alt. Clinical trials. Efficacy, or effectuor and sality of in ameliorating or curing or preventing that disease or related health. Controlled experiments, done to evaluate the condition. Clinical studies. Clinical drug development, defined as studying the drug in humans, is conducted in a sequence that builds on knowledge accumulated from non-clinical and previous clinical studies. Although clinical drug development is often described as consisting of 4 temporal phases, phases 1 to 4, it is important to appreciate that the phase concept is a description and not a requirement, and that the phases of drug development may overlap or be combined. The drug development process. Step 1, Discovery and Development. Discover new drugs through new insights into a disease process. Tests of molecular compounds to find possible beneficial effects. Existing treatments with unanticipated effects. New technologies. Conduct experiments to gather information on how it is absorbed, distributed, metabolized, and excreted. Potential benefits and mechanisms of action. Side effects or adverse events. Effects on different groups. Interactions with other drugs and treatments. Effectiveness. Step 2, Preclinical research. Can it cause serious harm, i.e. toxicity? In vitro, Latin for within the glass. In vivo, Latin for within the living. In silico, performed on computer or via computer simulation. Non-clinical studies. The development of a pharmaceutical is a stepwise process involving an evaluation of both animal and human __ information. The non-clinical safety studies, although usually limited at the beginning of clinical development, should be adequate to characterize potential adverse effects that might occur under the conditions of the clinical trial to be supported. Toxicology. Carcinogenicity. Immunogenicity. Pharmacology. Pharmacokinetics. Other evaluations and may encompass in vivo and in vitro models, and by modeling and simulation. Special populations may require additional non-clinical assessments. The clinical development process. Human pharmacology. Phase 1. Safety and protection of participants as the first priority. Healthy volunteers or in a selected population of patients with the condition. Estimation of initial safety and tolerability, dose tolerance, pharmacokinetics, pharmacodynamics and PK/PD. Exploratory. Phase 2. Investigate safety and efficacy, and refine effective dose, S, and regimen. Selected population of patients for whom the drug is intended. Randomized clinical trials of relatively short duration, dose-finding studies, biomarker exploration studies. Confirmatory. Phase 3. Confirm preliminary evidence that a drug is safe and effective, provide an adequate basis for marketing approval. Participants with or at risk of the condition or disease who represent those who will receive the drug once approved. Randomized clinical trials at a larger scale, clinical safety studies, studies in special populations. Studies of morbidity/mortality. Post-approval. Phase 4. Optimize the drug's use. And usually conducted as a regulatory requirement, FDA. More diverse populations. Comparative effectiveness studies, long-term follow-up studies, pharmacoeconomic and pharmacoepidemiology studies, observational studies. Interventions that can be evaluated by trials. 1. Drugs and other treatments of disease. 2. Medical and healthcare technology. 3. Primary prevention. 4. Behavioral interventions. Design of randomized controlled trials. Experimental study. Experimental study is also known as an intervention study with allocation of exposure, or intervention. Diagram showing randomization of a population into group with intervention and group with no intervention and determining outcome or no outcome from both groups. ASCII. Alt. Key elements in the design of controlled trials. Randomization. Why randomize? 1. Comparison groups, intervention and control, are made comparable with respect to potential confounders at the start of the study. 2. Helps reduce bias in the allocation of participants to the intervention or control groups. Alt. Define allocation ratio for treatments prior randomization. Example 1:1 allocation ratio, 50% receive new treatment, 50% control. Every treatment assignment has a known probability. Masking, blinding. Conceal treatment assignment to reduce bias related to prior knowledge or beliefs about treatments. Main steps in an intervention study. 1. Define the study question. Does the intervention affect the outcome? Alt. 2. Define the target population. Who will benefit the intervention? General population. Subgroup of the population. 3. Select the study population. 4. Allocate the intervention. Randomization is usually done by assigning random numbers to participants, allowing every participant a known chance of being allocated to any of the groups. Allocation concealment so that the assignment is not known. Is blinding similar to allocation concealment? Blinding may not be done in all trials, medical device trials. Allocation concealment done in all trials and done before trials. 5. Follow-up participants. See number 6. 6. Measure outcome. 4, 5 and 6. 1. Ensure completeness of follow-up. 2. Outcome ascertainment. Typically morbidity or mortality measures. May also be an intermediate outcome, which allows us to measure an outcome more quickly. 7. Analyze data. 1. Intention to treat, ITT, analysis. Analyze data based purely on randomization. 2. Per protocol analysis. Only those who completed the trial and who adhered to their allocated intervention. 3. Subgroup analysis. Small numbers, hard to determine that the treatment effect differs by subgroup. Unit of analysis. Were participants individually randomized to one of two groups? Were groups of participants randomized together to the same intervention? Were there multiple observations for the same outcome? If a vaccine has an efficacy of 80%, it does not mean that the vaccine will only work 80% of the time. It does mean that in a vaccinated population, 80% fewer people will contract the disease when they come in contact with the virus. ASCII. Alt. Vaccine efficacy. Percent reduction in disease incidence attributable to vaccination. Vaccine efficacy equals 1, RV, risk in vaccinated, /RU, risk in unvaccinated. Where RV/RU is the relative risk or rate ratio in vaccinated and unvaccinated people. Vaccine effectiveness. Sum of reduction in the clinical events that might be expected to be associated with the disease. Vaccine effectiveness equals unvaccinated rate, vaccinated rate/unvaccinated rate. ASCII. Alt. ASCII. Alt. Hazard ratio. Descriptor of how many times more, or less, likely a participant is to suffer the event at a particular point in time if they receive the experimental rather than the comparator. ASCII. Alt. Kaplan-Meier plot. Descriptor of how many times more, or less, likely a participant is to suffer the event at a particular point in time if they receive the experimental rather than the comparator. Alt. 8. Interpret results. Was there adequate control of potential confounders? Could bias have influenced the results? Was there blinding? Was analysis done on an intention to treat basis? ASCII. Alt. ASCII. Alt. Ethical issues in intervention studies. The investigator cannot allocate an intervention believed to be harmful, nor withhold an intervention believed to be beneficial. Hence trial is only ethical when there is equipoise. The Nuremberg Code's ethical guidelines for research. 1. Voluntary consent is essential. 2. The results of any experiment must be for the greater good of society. 3. Human experiments should be based on previous animal experimentation. 4. Experiments should be conducted by avoiding physical/mental suffering and injury. 5. No experiments should be conducted if it is believed to cause death/disability. 6. The risks should never exceed the benefits. 7. Adequate facilities should be used to protect subjects. 8. Experiments should be conducted only by qualified scientists. 9. Subjects should be able to end their participation at any time. 10. The scientist in charge must be prepared to terminate the experiment when injury, disability, or death is likely to occur. Code of Ethics. 1. The Belmont Report codifies basic ethical principles that underscore biomedical research. 2. The Declaration of Helsinki is an international agreement put forward by the World Medical Association and amended many times over the years to address new ethical concerns. 3. The Nuremberg Code was ratified following World War II, in response to the abusive and exploitative clinical trials undertaken by Nazi scientists. It provides an international standard for clinical research ethics. 4. The U.S. Common Rule ratifies key protections for the patients who participate in clinical trials. The role of Data Safety Monitoring Committees. ASCII. Alt. ASCII. Alt. What should control group receive? 1. Placebo, that is a treatment that resembles closely as possible the active treatment, but has no active component. 2. Standard treatment, where an existing treatment of established value for the condition in question is already available. Special design. Factorial design. A factorial design can be used to test more than one hypothesis in an intervention study. Outcome data and randomization of treatment A. ASCII. Alt. Crossover design. A crossover design allows each subject to receive the intervention treatment and the control treatment at different times during the trial. ASCII. Alt. Cluster randomized design. Entire groups of participants are randomized to different interventions. ASCII. Alt. Adaptive design. Allows pre-planned modifications to a trial based on analyses of accumulating data. Alt. Translational science.